Welcome back. I'm delighted to introduce our first plenary panel titled Genetic Preselection and Trial Design. This panel will include both talks and discussion, and we again encourage you to ask questions of the panelists during the talks and the discussion on Slido and in Slack. I'll turn it over now to Mark McCarthy, who is a staff scientist and executive director of human genetics at Genentech and a member of ICDA's organizing committee to introduce the panel.
Hello, good morning, good afternoon, good evening, wherever you are. Great to welcome you to this panel discussion where we'll have a series of talks and then hopefully plenty of time for vigorous discussion. I'll let the various members of the panel introduce themselves as they go through their presentations, but I'm Mark McCarthy from Genentech. And then we'll follow by chats from Sally, Unnur, and Kristin, and then Judy will moderate a discussion that follows it. So just to endorse Rachel's comments, please do add your questions to Slido and to Slack, and Judy will do her best to bring those together in the subsequent session. So the starting point for this panel and the efforts we've been making over the last few months to tackle this question comes from this recommendation that was included in the ICDA white paper earlier in the year around advancing genomically informed clinical trials. And the exhortation there that ICDA should work with scientists in industry, academia, and beyond to think about ways in which we can bring genetics and genomics into play in the design of randomized controlled trials. So we assembled a group of interested folks who are represented on this panel, but I should also call out to Adama Ibrahim from Novartis and Katrina Loomis from Pfizer, who also contributed to many of our discussions. And we've been, over the last few months, trying to wrestle this question to the ground. We've prepared or are preparing a document that hopefully will be disseminated in the early part of 2021 and which we'll submit for publication to summarize some of our findings. But a bit of a spoiler alert, which is that I don't think we've got answers to every question yet. So we don't have answers, but we have raised a number of very interesting questions in our discussions, and those I think we'll elaborate on over the next half an hour. It really is the case that we're raising this topic in the ICDA because we really do welcome some additional input from folks. So if you disagree with what we've said, or if you have a perspective that you don't think we're capturing, do reach out to us through any of the usual channels, and we'd love to include you in our further discussions. So, of course, there are many ways in which genetics and genomics is taking an increasingly important role in the whole drug development and discovery pathway. Obviously, you're familiar with the idea of genetics as a way to identify targets and define the indication space. Familiar with the idea that genetics can help us identify biomarkers that could support clinical development through Mendelian randomization approaches, for example. You'll be familiar, of course, that there are genetic variants in important metabolizing and other enzymes that can have a pharmacogenetic effect and have the potential to improve risk-benefit ratio. And also, you'll be familiar with the idea that many pharma companies now routinely perform genetic analyses within their randomized controlled trials across the whole population, looking for genetic predictors of therapeutic response or adverse effects. So those are all important, and those are all things that are well appreciated and in play. But what we're focusing on here is a particular question, which is to do with genetic preselection and the idea that this might be a way of generating more efficient clinical trials. Why might that be? Well, there are really two main reasons why you might want to do genetic preselection to inform your clinical trial. And these are really two extremes. There'll be many situations which lie in between. But on the one hand, you might simply want to do genetic preselection for reasons of expediency. Your assumption is that the drug that you're developing will eventually work on everyone and will eventually be released to everyone. But when you're in the early phases of development, particularly in phase 2, where you don't want to embark on a really expensive, long-winded study, and you want to get proof of biology and sort out dosing issues fairly quickly, you really might be tempted to want to accelerate that process by selecting those with higher event rates, so you can prosecute your phase 2 study with fewer patients over a shorter interval. That builds confidence then for a subsequent registrational trial that would not have similar selection, and then the presupposition is that the genetic selection would not be on the label. But the other situation is one where you really believe, at least first, that the drug will only work on a subset, or it will work best on a subset of patients. So you want to demonstrate that in your phase 2, and the expectation is that you will carry forward that selection into subsequent registrational trials, and ultimately that the label for the drug would restrict usage to those in the selected subgroup, which has its advantages, but also has some disadvantages, of course, because you're not really testing the idea that the drug might also be beneficial to those that are not in the selected subgroup. Trial preselection, of course, is not a new thing. In some situations, like rare diseases and cancer, we already use genetic preselection. A good example in the rare disease space would be the CFTR variants that Vertex used to select patients that went into the trials for some of their earlier range of medications. And of course, in cancer, it's quite normal to use tumor sequence variant information to target medications such as BRAF mutations and so on. We also do preselection in many situations with trials for common late onset diseases, but we generally, hitherto, haven't used genetics for that. But we've used things which are often connected to genetics, like circulating levels of a protein or cholesterol, for example, or we might use clinical state, such as embarking, at least initially, on a trial of secondary prevention of coronary artery disease to enrich for people who are more likely to have events. And the big question here really is whether we can move common late onset disease to a place on the left of this figure, and that's most likely to come through use of polygenic scores. So as you all know, we increasingly have robust polygenic scores that we can use to identify people who are at high risk of future disease. Can we use this to pre-select individuals who would recruit into phase 2 or phase 3 trials? Whoops. Seems a great idea, right? Why wouldn't you want to do it? But when you look at it, there aren't that many examples of this actually being prosecuted yet in pharma. And when you boil down to it, there are a number of reasons for that. And these are the reasons that are going to be expanded in subsequent talks. But just to break them down very briefly, there are some scientific reasons, and those are to do, for example, with issues to do with the standardization and portability of polygenic risk scores. Also possibly just down to the fact that one may be able to do preselection pretty well without doing genetics, simply by using something that's correlated with genetic levels, as in the case of hypercholesterolemia. Also concerns about, if you really go for high PRS individuals, are they really going to be representative of everybody who's possibly going to respond to the drug? There are logistical reasons. Those have to do with how do you identify the people who've got a high polygenic risk score in the first place, because we don't have universal genetic testing at the moment. We certainly don't have a standard of care diagnostic test in the polygenic space for many of these, unlike the situation with rare diseases and cancers. And if you don't have that, and if you don't have a set of people who are already defined as at high risk, you're going to have to deal in your trial preparation with all the issues to do with counseling and insurance and other related issues that will not have been tackled by the healthcare system to date. And then, of course, there are a bunch of commercial and regulatory reasons, some of which I've mentioned, but the challenges of having a genetic co-diagnostic, which certainly complicate the registration and regulatory process. And those questions about, is it a good thing or a bad thing to restrict the patient pool in a way that may identify people who benefit most, but might deprive treatment from others who would also benefit. So those are things we're going to discuss over the next 40 or 50 minutes. You may say, actually, these are problems now, but these are problems that are going to get solved. There will be more rollout of universal genetic testing. We will solve some of these issues to do with polygenic risk scores and their portability, and that's true. But one thing just to bear in mind is that the clinical development plans that are currently being made in pharma are for drugs that may not be licensed till the end of the decade, for example, given the time that it takes. So these are decisions that pharma companies are starting to have to make now, because the drugs that they're developing may come out into a world where many of these things have been solved, and we need to be thinking about ways in which we can ensure that the drugs that are being prosecuted at the moment, the targets that are being prosecuted at the moment, are well suited for the world into which they may emerge. So with those opening comments, I'm going to pass this on to Sally John.
Thank you, Mark. So I'm going to talk, I guess, about some of the logistical considerations for clinical trials, even for rare diseases where the scientific case seems very obvious. I should say I'm from Biogen too. Thanks, Mark. So, when a team is thinking about a new clinical trial, the decision to implement genetics is really dependent upon the evidence base that the genetics can contribute to answering the question that's actually being asked as part of the clinical trial. And for many clinical trials, there is no genetic evidence that they will affect efficacy or safety. And I think industry's done a pretty good job at this left-hand end of banking samples. We now do routinely bank samples from clinical trials, which does allow us to do retrospective analysis when a hypothesis comes up or we see something in a trial. But what I'm going to talk about today is this case where we have a strong prior hypothesis that the drug is only going to work in a particular genotype group or will work better in a specific genotype group. So can I have the next slide, please, Mark? And to get going for, rarer diseases is the examples I'm going to talk about, specifically neuromuscular disease. But for any rare disease that may be scattered around a few cases here and there around the globe, we do need a better understanding of epidemiology, so the incidence and prevalence of disease, and natural history. What level of clinical heterogeneity do we see? Is there a big variation in progression and symptomatology of the disease? And the more solid and robust information have there, the better able we are to design a clinical trial to really test the effect. And in addition to that, we often find ourselves in a sort of catch-22 position where if there's no therapies, then there is no genetically validated test done as part of standard clinical care. So we often need to work out how we're going to find those patients with the genetic variants that we care about. In addition to that, you'll have heard as part of this meeting and the ICDA in general that we are finding more and more genes and more variants, and these are often not fully curated with respect to what's actually pathogenic and the penetrance of those mutations. So we need to start early, even for rare genetic diseases. So can I have the next slide, Mark? So I guess the first example could be considered a success story for the field, and that's spinal muscular atrophy, neuromuscular disease caused by deletion of the SMN1 gene. And this little figure on the left-hand side shows that there are subtypes of SMA which are determined by the severity of the disease, which is correlated with the number of SMN2 copies that patients carry, as the SMN2 gene still produces some residual protein. So I haven't got a pointer, but the green bar there is kids with type 1, and they're often diagnosed they are diagnosed in infancy. They often don't meet any motor milestones, and the natural history of the disease is well understood. So not surprisingly, the early trials for medicines were carried out in these symptomatic patients, where the patients were recruited based on a clinical diagnosis and a confirmation of both SMN1 deletion and the SMN2 copy number. Actually, can you click again? I think there's some animation I didn't know about. And again, thank you. Oh, no, back. Sorry. Yeah. Okay, so we had good preclinical data at the time, though, in animal models that the earlier one treated, the more likely you could rescue the model and therefore potentially see efficacy in humans. And so what the community really needed to do was to treat pre-symptomatic patients. And as there was no treatment, there was no standard testing of newborns. But there was an awful lot of work that went into this ahead of time. And the pre-symptomatic trial for nusinersen started in 2015. At the same time, scientists from Biogen contributed to a study that was led by the newborn screening division at the CDC, who published and were working on an assay that would be suitable based on a blood spot for newborn screening. The first drug was approved at the end of 2016, and in early 2018, SMA was added to the list of conditions for newborn screening in the US. Now we have 3 treatments approved for SMA, and now we have this newborn screening. This is a success story in that this is quite a quick time frame, I think, to get from a disease with no treatment to a fully implemented newborn screening and 3 treatments. So if we go to the next slide, now moving on to ALS, and this is a case where we are developing a drug that treats SOD1. We have a strong biological rationale that this medicine will work only in those patients that have a SOD1 mutation. ALS is, again, a relatively rare disease. It's a progressive neurodegenerative disease, motor neuron disease in the UK. About 5 to 10% of patients are familial, and of those, about 20 to 30% will have a SOD1 mutation. About 90% of the case have been called sporadic, saying that the cause of the disease is unknown, but actually now we know that about 5% of those patients will also have a SOD1 mutation. So the trial down there on clinicaltrial.gov is restricted to carriers of a SOD1 mutation, which seems fairly straightforward. However, there's still an awful lot of work to do. In addition to the familial patients, how do we identify those non-familial patients who are not having routine genetic testing for SOD1? In addition to that, even within the SOD1 gene, certain mutations lead to very aggressive disease and some to much less aggressive disease. And adjudication of pathogenicity is important for inclusion onto the clinical trial. So there's still a great deal of work to do, even in this very specific case of recruiting patients with a SOD1 pathogenic mutation. So if we move to the next slide, please, Mark. And this is a case where, going back to that evidence base, we're developing a therapy for ataxin-2, and this is also thought to be a genetic cause in a very small subset of ALS patients. But here we have strong biological data that this therapy would be suitable for all patients with ALS, not just the subset with the ataxin-2 mutation. And that's driven by the fact that it alters TDP-43 pathology, which is pervasive in ALS. And this is an example of where it turns out to be good to collect DNA in clinical trials. We were fortunate enough to have samples from the Empower study. This was a negative phase 3 study of dexpramipexole, and we had consent from a proportion of the patients on that trial for future exploratory use. When we looked, we had 7 patients on that trial with ataxin-2 repeats. And this isn't a scientific presentation, but you can see here that based on the functional rating scale, the 7 patients had a faster rate of decline. Ellen and Nick from Biogen did this work. We've also worked with academic partners, such that now when we think about this evidence base, we are sufficiently confident that these patients behave differently from patients that don't carry the repeat. And so the clinical trial design on the right of the slide demonstrates that we will recruit separate cohorts onto this study, cohorts who have non-genetic causes of ALS as all comers, but also a separate cohort of these ataxin-2 repeat carriers. So if we go to the next, so this is my last trial, and I think the messages that I wanted to come here, even for what appear to be rare genetic diseases, there is still a lot of work to be done and lots of questions to be asked, and building that infrastructure early to match the patients with the genotypes to the clinical trials is really critical. I think in some part due to the very rapid rate of novel medicines that are being developed now for rare diseases, there is much more activity in the community at large in trying to do this. And I've just pulled these things off the internet. Actually, IQVIA is a contract research organisation that runs clinical trials, is partnering with Genomics England. Invitae is a clinical diagnostics company aiming to identify patients with a genetic form of epilepsy. And so we see this recognition that if, as we are getting to developing medicines for rare diseases, that we do need the infrastructure to get those patients onto clinical trials. So that was my little section from the rare diseases, and I'm going to pass it on to Unnur now to perhaps tackle the more complex question of common diseases. Thank you, Unnur.
Thank you, Sally. So I'm gonna discuss if and how genetics of common diseases can actually enhance the efficacy of randomized clinical trial for common diseases. Next slide. there are a large number of sequence variants that have been identified that associate with common diseases. For them to have impact on individually, to have impact on risk stratification, they need to be at relatively high risk and be relatively common. There are, however, very few examples of such variant, one being the ApoE locus for Alzheimer. However, a large fraction of the variants that have been identified are common with small effects. However, by combining those, the effects of those variants into polygenic risk score, we'll start to give them some clinical utility. Next slide. Okay, in Iceland, we have built a database of genotypes and phenotypes for a large fraction of the population. And this data set we have used for target discovery and prevention, and we are reporting back some of the high risk actionable variants, such as the BRCA2, back to the population. And as of lately, we have started to use this source in the context of randomized clinical trials. Similar sources are also being built in Finland, UK, and US, which might have the same implication for clinical trials as the Icelandic population. Next slide. Using genetics, a sequence variant was identified in the amyloid precursor protein that was found to actually reduce the cleavage of the protein by beta secretase and therefore reducing the accumulation of a beta. Based on this genetic evidence, a base 1 inhibitor was developed that actually inhibits the beta secretase and thus thought to reduce the generation of A beta. I'm just going to talk briefly about a clinical trial using this inhibitor that was conducted by Amgen and Novartis. Next slide, please. So the study was at 24 sites worldwide, including in Iceland. And for increasing the number of events during the trial period, and therefore increasing the efficacy of the trial, participants had to carry at least one ApoE4 allele. ApoE4 is the biggest risk factor for Alzheimer, with an odds ratio of 12 to 15 for ApoE4 homozygotes. And these homozygotes, they start to deviate from the population regarding cognitive decline at the age of 55. Next slide, please. So instead of screening the population for ApoE4 carriers, we were able to use the encrypted genotype database at Decode Genetics in Iceland. And by screening about 170,000 genotypes individuals, we identified about 1,100 ApoE4 homozygotes and about 11,000 ApoE4 heterozygotes that were 60 to 75 years of age and without Alzheimer. I think this example highlights or underscores the importance of having access to large population-based genotypes in the context of clinical trials. All of the, or most of the homozygotes were actually recruited into this, or invited to take part in the study, and equal number of heterozygotes were also invited. Those who agreed to take part in the study were about 50% of those that were invited, and the ratio between the ApoE homo and heterozygote status was very similar. However, and unfortunately, during a planned review, worsening in some measures of cognitive function was identified and the trial was therefore terminated. Next slide, please. So now turning to the polygenic risk score. Like Mark said, polygenic risk score have been improving in recent years, both in their predictive value and generalizability. And this is mainly due to the large GWAS studies that that produce more accurate estimate for the effects of the variants. And there are also improvements in computational methods in the generation of the score. The score clearly can identify individuals at high and low risk of disease. And this is exemplified here for the coronary artery disease and MI, where the odds ratio for those who are in the top 2.5% of the polygenic risk score for early onset CAT is 3.2, and for early onset MI 3.5. And these are odds ratios that are similar to the rare familial hypercholesterolemia variants. The risk score can also be very valuable in identifying individuals before they are presented with traditional risk factors, and also for diseases that lack measurable risk factors. Next slide, please. So this is a slide from using data done in UK, and this shows that for those who are in the top 8% of the polygenic risk score, they are enriched for traditional risk factors for coronary artery disease, but they do not explain all of the risk. Next slide. So in Iceland, we decided to do a prediction model to evaluate whether actually polygenic risk scores could improve the prediction of cardiovascular events. And we used traditional coronary artery disease risk factors, and the polygenic risk scores that we used were for hypertension, stroke, CAT, and MI. And we also added into the model 4,900 plasma proteins. Next slide, please. These graphs show 10-year prediction of a cardiovascular or event-free cardiovascular event for those that are in the top 5% of highest predicted risk. And the blue line is the baseline for the traditional risk factors, and the orange one is with the addition of the polygenic risk score, and then the green with the addition of the proteins. As can be seen, it is clear that the polygenic risk score does improve the prediction, especially for MI, and this can also be seen for the area under the curve for event within in 5 years. So in conclusion, the advantage of genetics in the recruitment of design of randomized clinical trials is going to be beneficial, especially in the light of the growing number of populations that have large population-based genotype data set that will be ideal grounds for performing genomically informed clinical trials. Furthermore, having genotype data on all participants will also allow for various post-hoc analysis. Thank you all. So next speaker is, okay.
Thank you. I'll pick it up from here. Hi, everyone. I'm Kristin Van Goor. I'm the Senior Director of Regulatory Policy and Intelligence at Vertex, and I'll be focusing on the regulatory considerations for genomically informed clinical trials. Next slide. So the good news is there is an FDA guidance that addresses the topic of enrichment that has direct applicability to what we're discussing. So the previous presentations did a very nice job of providing some specific examples, both in the rare and common disease spaces. where we've thought about enrichment strategies, and the FDA guidance on enrichment very much aligns with some of the concepts, ideas, and concerns that arise in those scenarios. I will say too that this guidance is not specific to rare diseases or common diseases. It really does have applicability across the disease spectrum. So while the guidance focuses for the most part on using enrichment to improve the ability of a study to detect drug effectiveness, FDA says that these strategies should also be or could also be employed to assess safety. And FDA's goal in issuing this guidance is to provide recommendations that increase the efficiency of drug development and also support precision medicine. So again, very much consistent with what we've been talking about today in this session. Next slide. So to go to the regulatory concerns, these are the things that are top of mind for FDA when they're thinking about enrichment strategies. The first one being generalizability. So their principal concern, and this is true for any clinical trial, is whether or not the clinical trial population is really representative of the population that's likely to use the drug or may benefit from the drug in the real world. And so when you add enrichment on top of that, that concern about generalizability comes very much into play. The second thing that FDA is thinking about is whether or not the enrichment strategy that's employed during research in the clinical trials is a strategy that could be used in practice. So again, they're really thinking about the real world scenario. Is this something that you can measure in the course of clinical care? Is the testing strategy feasible? And to the last bullet, is the testing strategy reliable? So the accuracy of the measurement, the sensitivity and specificity of the test is going to be critically important to not only the success of the enriched clinical trial, but the ability to understand and predict the proper use of the drug in the real world. Next slide. So there's a balance, and FDA talks about this balance and urges drug developers to think about finding the proper balance when they design these studies. So on one hand, you can decrease heterogeneity, you can increase your study power through enrichment, but at the same time, if you narrow down too far, you're potentially ending up with a study that's providing too little information about the full range of patients who may receive the drug in clinical practice. And so FDA reminds drug developers that this needs to be very carefully considered during the design of the study, thinking about those subgroups that may have a need for information about use of the drug in the future. Next slide. FDA provides some guidance on both prognostic and predictive enrichment, and I think this has been covered really nicely in the previous presentations. But on one hand, prognostic enrichment is going to increase the likelihood of seeing the endpoint that you need to see in your clinical trial, or see worsening of the condition in the course of a shorter duration study. So really getting to a smaller sample size, a higher likelihood of seeing the events that you need to see to understand what your drug may be doing or if it's working. On the predictive enrichment side, this is where you're identifying patients that are more likely to respond because of genetics and a relationship to the mechanism of action of the drug. Again, improving study efficiency, feasibility, and enhancing the benefit-risk relationship for the patients in the subset. Next slide. So when FDA thinks about where enrichment comes into play throughout the drug development process, they point to a number of areas. One, in those early studies of efficacy, so that you can get to proof of concept and inform the design of later studies, which are often your larger studies. In the second scenario, and we've touched on this a bit earlier, is where you have a drug where you may be seeing some significant toxicity, a lower overall response rate in the broader population, but through identification of a responder population, you could get to a place where you are able to continue the development of that drug in the subset and get to a successful development program and potentially regulatory approval. So FDA reminds that a trial intended to provide evidence of effectiveness to support approval could include a broad range of patients, but be prospectively designed to evaluate in its primary analysis the effect in the enriched population subset. Next slide. The other option that FDA provides is an empirical genomic strategy. So while many genomic markers have been linked to pathophysiology property, the linkage is not necessarily essential. So FDA acknowledges that you could run your trial and then identify a subset of responders through analysis that takes place after the original trial. Next slide. And so now we circle back to regulatory cautions and interpretation, and this relates very much to the points that I began with. The validity of the test is really critical, and FDA says this a number of times in the enrichment guidance, even though it is not a guidance about analytical validity and companion diagnostics. So FDA is reminding drug developers that thinking about how you're going to establish your test, understanding the biomarkers, clinical sensitivity, specificity, positive and negative predictive values, all of these things need to be very well characterized. And they say it quite bluntly in the guidance. An inaccurate assay will undermine an enrichment effort. So that's something to always keep in mind. And then finally, FDA talks about the fact that enrichment strategies should be clearly described in the study report and the labeling to indicate how the drug is to be used and to whom the results may apply. And this is an important point that perhaps we can get to a bit more in the panel discussion about how the design of the study really puts you on a particular path for not only the labeling of a drug, but the potential need for a companion diagnostic, which has clear and direct downstream effects on access of patients to the medicine. So with that, I think it brings us to our panel discussion.
Thank you, Kristin. We have a number of questions from the audience. Rory Collins takes issue with the use of expediency as a way of thinking about trials, and it's better to make trials less costly and complex, focus on RCTs, and take advantage of technological advances. Does anyone want to comment to those comments, Dr. Collins?
Yeah, I use that term. I don't think it's an accepted one, but I think it and yeah, maybe there's better ways of framing it. And there was the prognostic versus predictive is another way of explaining the same difference. But I think it still speaks to the idea that you are trying to identify a high-risk group in whom you can get to important answers about, particularly about biological confirmation or side effect profile in a more expedient way, less complex, less expensive, less prolonged way, so that essentially you can get the medicines into the right patients sooner, which is the goal of everything that we're trying to do.
Does anyone else on the panel want to comment on that? Okay, so the next question is for Kristin. It's from David Goldman. Does the FDA have a specific panel, expert panel, that focuses on this, on how predictive and well-validated pre-selection should be?
So not a panel of experts necessarily, but FDA does have guidance that's been issued on the development of in vitro companion diagnostic tests. So there are well-established expectations for the validation of a companion diagnostic.
Thank you. Next, we have a question from Tim Fraley about the ethics of being invited to a trial with respect to the ApoE4 mutation. Unnur, do you want to take that on?
Yeah, maybe. So the recruitment, maybe if you remember my slide, they were also invited to take part in the study individuals that were not ApoE4 carriers. So it wasn't set out to be designed like that, but there was they were actually represented larger fraction of those recruited. However, as part of the trial, once they were in the trial, one of the first steps was to actually, they had to agree on getting their ApoE4 genotypes. So the DNA sample was taken and they were genotyped again, and only if they accepted to get their ApoE4 genotypes, they were allowed to take part in the trial.
Thank you. There's a question.
Can I say
Sure.
something on that? I mean, I think it's a really good question, and it's certainly something that exercising something. You know, people will know there's a study planned in the UK of 5 million people getting polygenic risk scores, and clearly one of the great things that could be done there is to identify people who've got particularly high polygenic risk scores, you know, for diseases like Alzheimer's, but that's not the only example. And clearly understanding how that is going to be addressed and the ethical and other issues that go with that is going to be a very important part of making sure that one can, in the right way, access those populations, which you really need for many of the studies that we want to do, both observational and interventional. I think there has been, I can't quote you on this, but I think Sally sent me this, you may remember, that there has been some focus group and survey done that in general people are accepting of the idea of understanding their Alzheimer's polygenic risk and favorable, not everybody, but you know, there's certainly a good corpus of people who are insufficiently interested and would like to know their ApoE or polygenic risk status, even in the absence at the moment of obvious interventions that would be consequent on that.
Yeah, I can add to that, because I think in the rarer disease space, it's often usual to have genetic counselors available. The genetic diagnostic companies provide genetic counselors, and I guess we need to work out how we support people in the more common disease space with high risk rather than necessarily more deterministic type mutations.
Thank you. There's another comment from Rory Collins, again for the entire panel. Prognostic enrichment may backfire if benefits of treatment take time to emerge, and it also reduces safety assessment. Does anyone want to react to that?
You want to start, Mark?
No, I think these are all part of the balance. I mean, I think for all of us who embarked on this and started thinking about this, on the one level, it does seem what I would say is a no-brainer. You think we all understanding the power of polygenic risk scores to predict. We all understand how expensive it is to develop drugs. We all understand how important it is to try and have more efficient clinical trials. So it does seem that the coming together of those is something that should be celebrated. When you actually get down to the detail of what it actually would take to make that policy instantiate them, you run up against some of the concerns that we've raised, and I think you've raised another one there. So it's all about establishing the balance. And as I said, this is something which is very much a moving target at the moment, part because some of those obstacles, particularly the logistical ones, will get solved as we get presumably more rollout of genetic testing across the population. Some of the issues to do with polygenic risk scores will evaporate as we get better at having stable and portable polygenic risk scores. So it's an interesting space because there are a number of significant obstacles, and we're going to have to understand how those play out, and also understand how they what the impact is on the regulatory environment.
Thanks.
I guess I could. I do feel that the natural tendency is not to do enrichment, probably for the reasons that Rory raises, that it does bring its own issues, and I think some of it might be due to can we get better at the generalizability? So if you have a polygenic risk score, can we show that apart from the genetics, in lots of other ways, you are generalizable to the rest of the population? And I think the sort of compromise that often happens, whether it's genetics or other potential enrichment factors in clinical trials, is an exploratory endpoint is written into the clinical trial. You recruit everybody and then you do some kind of stratified analysis later, which is, I guess, the best of both worlds, but expensive. And I do think thinking about generalizability, because Kristin, you may want to comment on this, is important, because you are going into the real world, and you know your medicine will be prescribed to a large number of people, so you want to be confident that it is relatively generalizable to the population who may be prescribed it.
Right, Sally, I think this is really important point, because, as Mark says, the studies that are being designed right now are for drugs that may be approved in 2030. So there's a lot of lead time on this. And it's the design of the study, it's the exclusion criteria that you put in place that really determines who will have access to the drug, right? So if you believe that the drug may work in a much broader population, but you want to run your trial with a very focused, enriched subset, you do need to have a plan in place to start to generalize your findings. Otherwise, you will run up against years later looking back at those clinical trial inclusion-exclusion criteria and having that be essentially the basis for your label, which becomes the basis for access to treatment. And so it is a really important point. I think, as FDA and other regulators get more familiar with and accustomed to use of real-world data, there's potentially some good data sources in the post-approval setting that can be leveraged to start to fill in those gaps or make the bridge from the clinical trial population to the broader population. But I think the fact remains is when you're designing the study, you have to think about how you're going to get there, whether it be through study of a broader population and doing a subset analysis, doing an expanded, larger phase 3 study, or doing post-approval data collection.
Thank you.
I think I want to make it maybe say one thing, that okay, people are being recruited into clinical trials based on traditional risk factors. What is the difference between using polygenic risk scores that are actually sort of reflecting some of the traditional risk factors?
Yeah, I mean, you raised the question.
Yeah.
If you do a trial selection on LPA, for example,
Yeah.
you could do that on little LPA levels, or you could do it on genetic predicted LPA. They'd be very highly correlated, but perhaps the perspective on both of those trials would be somewhat different, and there's no real scientific logic for those to be viewed differently.
So a major question we've gotten is from Anonymous, which is, how do you weigh the ethical issues around possibly increasing health disparities using PRS that don't translate well across populations?
Hugely important question, and something that I think everybody is aware of. I'm optimistic that the community is aware of this, and ICDA and others are doing their very best to improve the situation, both through focusing collection of biobanks from populations that have not been well served historically, but also by some of the improvements in statistical methods that mean that is improving. Now, I don't think that's a problem that is peculiar or specific to polygenic risk scores. I mean, a lot of our medicines are developed in North America and Europe in well-developed societies and shown to work there. And there are many reasons, not just polygenic risk score, but the spectrum of rare variation or other environmental factors that can influence their impact. So at the end of the day, this is a broader question than just the polygenic risk scores, one that's got to be solved, but it's not one that's specific to this issue, I would say.
Thank you. Question for Unnur from Ricardo Verdugo. In the CVD trial, PRS positive proteins increase AUC in 2%. Would that be enough for Iceland's health authorities to include it in critical clinical practice guidelines?
Well, I'm not sure.
I think that's fair.
Yeah, it's. I think there are a number of issues that need to be considered, and I think everything that is gonna. I don't think that Icelandic health authorities would really. It would be more like the use of the genetic data, if that would be available to studies like that.
And I think there are other settings. I mean, cardiovascular risk, we're quite good at predicting through traditional, inverted commas, risk factors. I think if you think about something like Alzheimer's, which we are very poor at predicting, and we need to be able to predict earlier if we're going to be able to influence the disease progress, which looks to be what we need to be able to do with Alzheimer's medication and other interventions, then that's going to be the sort of situation where probably the genetics is going to have a bigger impact. Because
terrific. A question for Kristin from Miles Axton. Does the FDA specifically advise differently on empirical biomarkers established during pre-treatment phase or during treatment in a trial?
That I don't know offhand. I don't recall seeing any specific recommendation on that, so that's an area where I would imagine you'd have to go in for advice specific to the program.
Okay. Let me take one more question. There's actually a series of questions, so I apologize that we can't get to them all. But I think this is actually an interesting question from Abhi Ratnakumar. Is there much work using genetics in combination with sex-stratified RCTS? And why don't we take that as our last questions?
I'm not aware of anything. I mean, obviously, most polygenic risk scores seem to work pretty well in both males and females, but there are one or two examples where that hasn't been shown. But I'm not aware of any examples of that.
Is anyone else aware?
Not
All right.
off the top of my head.
Yeah. So, Rachel, should I hand it back to you?
Yeah. Thank you all so much for a great and really interesting discussion. Thank you to Judy for moderating our discussion this afternoon where I live. We'll now move into a break of about, I think, 13 minutes. We're just about on time to end at, the break will end at 8 o'clock p.m. UTC. We know it's getting kind of late for folks in Europe, so we're really grateful that you're staying with us. And thank you so much. We'll get started with Plenary 2, Mechanisms in Medicine then.